All right, let's put on our detective hats. Today, we're diving into a massive genetic mystery, one that's at the very heart of biology. How does evolution really work? I mean, we know it's a thing, but what's happening behind the scenes, at the genetic level? To crack this case, we have to start with a huge question that basically shaped the entire field. So here's the big question that for a long time really split the scientific community in two. Is evolution the super slow, gradual crawl that takes millions of years to show any real change? Or is it more like a series of sudden, giant leaps that can create a whole new species in a flash? This was a major debate. And this whole debate really came down to two key figures. You had, of course, the man himself, Charles Darwin, but then you had this other guy, a botanist named Hugo de Vries. And based on his own research, he came up with a totally different idea for how this whole evolution thing worked. And this is where you can really see just how different their ideas were. Darwin's view was all about small, tiny variations adding up over time. It was a gradual, directional process, always pushing a species to be a little bit better adapted. But de Vries, he thought the real driver was these big, random, totally directionless mutations. He even had a name for it, saltation, this idea that one massive mutation could create a new species, bam, just like that. So for Darwin, it's a slow march. For de Vries, it's a sudden jump. So who gets the prize? Was it the slow crawl or the giant leap? Well, as it turns out, the real answer was a bit more complicated than either of them probably could have guessed. And to figure it out, we needed a whole new field of science to step in: population genetics. You know, if you want to measure whether something is changing, you first have to know what no change looks like, right? It's like trying to tell if a car is moving without seeing any trees or buildings standing still next to it. So, population geneticists came up with this brilliant idea, a model for a population that is not evolving at all.
And that brilliant idea is called the Hardy-Weinberg Principle. Basically, it says that the frequency of alleles, the different versions of a gene, like the gene for blue eyes versus brown eyes, will stay the exact same generation after generation as long as there are no evolutionary forces messing with it. This gives us the perfect baseline, a fingerprint of a non-evolving population, to compare everything against.
And when a population is in this state, we call it genetic equilibrium. You can think of the gene pool as just all the genes and all their different alleles in an entire population, all mixed together. So in this perfect Hardy-Weinberg equilibrium, that entire pool of genes just stays completely stable over time, no changes. Okay, don't let the math scare you. It's actually super simple. If P is how often you see one allele, let's call it big A, and Q is how often you see the other allele, little a, then this equation just predicts the mix of individuals. P squared is the frequency of getting two big A's. Q squared is the frequency of getting two little a's. And that 2pq in the middle, that's everyone who has one of each. And since that's all the possibilities, it has to add up to 1, or 100% of the population. And this is where the real detective work kicks in. When scientists go out and measure the actual gene frequencies in a real population, and those numbers don't match what the equation says they should be, bingo, they've found their proof. The difference between what they expected and what they actually found shows that the equilibrium is broken. It's the mathematical smoking gun that evolution is happening. Okay, so if a real population isn't in that perfect, stable equilibrium, what's knocking it off balance? What are these forces that are disturbing the peace? Well, scientists have zeroed in on 5 key agents of change that can totally disrupt that Hardy-Weinberg equilibrium. Let's meet the usual suspects. And here they are, our 5 main culprits. You've got gene flow, which is basically just migration, genes moving from one population to another. There's genetic drift, which is change that happens just by random luck. Mutation, which actually creates brand new alleles. Genetic recombination, which shuffles existing genes around into new hands, kind of like a deck of cards. And last but not least, the big one, natural selection. Let's talk about genetic drift for a sec, because this one is fascinating. This has nothing to do with survival of the fittest. It's more like survival of the luckiest. It's just random fluctuations. Picture a small group of beetles. Just by pure dumb luck, more of the green ones happen to get stepped on than the brown ones. The next generation is going to have fewer green genes, not because brown was somehow better, but just because of chance. And this effect is way more powerful in small populations.
A really classic example of this genetic drift is something called the founder effect. This is what happens when a tiny group breaks off from a bigger population and goes to start a new colony somewhere else. Just by chance, the genes in that little group of founders might be really different from the original population, and boom, you set that new population on a totally unique evolutionary journey. So, while forces like drift and mutation are pretty random, natural selection is the complete opposite. It's the one force that isn't random at all. It takes all that random variation that's been created and starts to act on it. It's what gives evolution a direction, favoring the traits that actually help an organism survive and reproduce. It's the mastermind that shapes the population over time. And natural selection can work in a few different ways. First up, you have stabilizing selection. This type favors the average Joe, the individuals right in the middle. It actually selects against the extremes. A great example is human birth weight. Babies that are either too small or too large tend to have lower survival rates, so selection pushes the average right toward that sweet spot in the middle.
Next, we have directional selection. Now, this is what most people probably think of when they hear evolution. This is when one extreme trait is favored and it causes the whole population's average to shift in that direction over time. Think of the classic giraffe example. Having a slightly longer neck meant you could reach more leaves, so selection favored that trait. And over generations, the average neck length of giraffes got longer and longer.
And finally, there's disruptive selection, which is maybe the coolest one. In this case, individuals at both extremes have an advantage over the ones in the middle. Imagine a bird population where there are only two types of seeds available, really big ones and really small ones. This would favor birds with either huge beaks or tiny beaks, while the poor birds with average-sized beaks can't efficiently eat either type. So let's pull this all together. That old debate, Darwin versus de Vries, it turns out they were both kind of right. You have these random forces, mutation, genetic drift, that are constantly creating new variation, just throwing stuff at the wall. And then you have the non-random force, natural selection, that comes in and decides what sticks. It's this amazing dance between chance and necessity that really drives evolution.
And that kind of leaves us with one final big thought. With all these powerful forces of change constantly swirling around, migration, mutation, chance, selection, is it even possible for a population to be in that perfect Hardy-Weinberg equilibrium out in the world? Probably not. And that's the beauty of it. The principle was never meant to be a description of reality. It's a theoretical tool, a perfect baseline that allows us, the genetic detectives, to finally see and measure the beautiful, messy, and constant process of evolution all around us.